record, and then I can share. Hopefully it will mess it up.
Yeah, okay. Well, let's get started then. Oh, yeah, I want to introduce Gabriela. Gabriela Imre works at the core facility, at our core facility, since 10 years, 11 years, something like this. And she is super good with sample preparation. So please, Gabi.
I'm lucky enough that a lot of things that I will talk about were already mentioned, which means that now they will really, really settled, hopefully. So I'm happy to be able to give this lecture. So let's get started. Moment. So we all have different samples or starting point for our staining. We can work with cell in monolayers, organoids, tissue sections, and some of us even work with plants. And all of us want to end up at the microscope to image our sample. And there are many steps we have to take till we end up there, including the immunostaining. But in this particular lecture now, I will talk about the steps before we start the immunostaining and some steps right after the immunostaining. So what I will talk about in this sample preparation is what the type of the sample carrier, what you should choose and why, where you should place the sample. We will spend some time talking about the fixatives, autofluorescence, which can, take care of it either before you start the immunostaining or after, and which mounting medium you should use, and the different types of mounting medium. So let's get started with different types of sample carriers. So the sample carrier, there are many different types of sample carrier that you can use, and the manufacturer are continuously trying to figure out and find the best sample carrier that might be good for your application. They make new and new. But the choice of the sample carrier will depend on the type of sample that you want to that you have, also on what you want to do, what type of experiment, and how you want to analyze. So before choosing a sample carrier, it's quite important to ask yourself some questions, or decide, well, I work with cells or tissue organoids, because depending on your type of sample, your choice will be different. Do I image live or I image fixed samples? What is the resolution I need to answer my scientific question? Do the cells grow on glass? ECF is perfect, but some of them are very unhappy on plastic, so you might need to use plastic. And if it's plastic, should I use a thin or a thick plastic bottom sample carrier? And how many samples would I have? Do I have 25, 35, 46, or I only have 2 or 3 samples I want to image? And of course, more importantly, if I can you use the sample carrier on the microscope available in your lab or in the facility? I mean, it's really annoying because I don't know how to get used. So what should
You
you
have
pay
the
attention?
3 little dots. Okay, you managed.
Yeah. So what should you pay attention to when choosing your sample carrier? Well, you already heard it before, and I say it again, is the thickness of the cover slip, or the thickness at the cover slip at the bottom of your multi-well plate, dish, multi-well chamber. And our recommendation is number 1.5 glass. Number 1.5 means 170 micrometer thick, with a tolerance of plus minus 10 micrometer, meaning 160 to 180 micrometer. If you do perform super resolution, then you would choose a number 1.5H, which has a thickness of 170 micrometer with a tolerance of plus minus 5 micrometers, meaning 165 to 175 micrometers. So what are the consequences if we are using cover slip that are not number 1.5? So you will create chromatic aberration, so that it will lead to a degradation of the image quality. It will affect the brightness, but it will reduce the intensity of your signal. It will affect the flow focus. You will have a hard time to focus properly, so image might be blurry. And the high numerical aperture objective are highly sensitive to the thickness of the cover slip. So here I have an example where I managed, it was not easy to find cover slip equal number 0, number 1, and we had our own number 1.5. And I took some 200 nanometer beads, which are, I put them on the cover slip. And after that, I went to the microscope and I was imaging with a 60x oil objective, which has a numerical aperture of 1.4. And for all the images, I had exactly the same imaging settings. I did my absolute best to focus in the same way, to be on the same focal plane. And I displayed the images in the same way. And then I display the images and I'm looking to the intensity of each of these beads, which are exactly the same. You can very well see that the intensity drops as we are the thickness of the cover slip changes. So my recommendation to you is to do your best and always use number 1.5 is possible glass. But of course, some cells might not attach well on glass. Or if they do, they might change morphology than growing on glass. It happens, and it's not very uncommon. So what should you do? Well, you can choose actually to image using cell culture multi-well plates or dishes. But you have to keep in mind that the bottom is thick, it's more than 1 millimeter thick, often autofluorescence, and definitely not flat. So you will have a very hard time to keep the focus over large areas. But what can you do? This is what you cannot do anything else. So if your application requires the use of thick plastic bottom carriers, always use long working distance objective or extra long working distance objective, which will allow you to image through the thick plastic. And only use these thick plastic bottom dishes or multi-well plates if you are sure that you do not need high resolution imaging. In case you do need a high resolution to answer your scientific question, use multi-well plates, dishes or chamber with thin optical plastic bottom. They are also labeled as number 1.5 and they have the same thickness, 170 micrometer. Now that you choose your sample carrier, you say, oh, this is what I would like to use. It works perfectly for my sample, my scientific question and resolution. It will do it. Then you have to go and check if it actually works with the available microscope. So here I depicted an upright and an inverted. For fixed samples that I placed between the slide and the cover strip is no difference between which of the microscope you will use, if it will be an inverted or an upright. But in the moment that if you choose to work with a multi-well plates or multi-well chamber or small dishes, your choice should be the inverted microscope. Because if you choose an upright and you try to focus, your objective will bump into your sample carrier without even having the time to reach the correct focus. For intravital imaging, have that little mousey there, the upright microscopes are the best. What to pay attention when you buy your favorite sample carrier? Let's type to the slide, which is also a sample carrier. It is actually, and it used quite a lot. It's very important that you pay attention what kind of slide you are buying. There are different types. They are uncharged, they are charged. They are most of what we want, especially if we are working with tissues, that we have a positive charge on our slide. So the tissue is binding very well to the slide because the tissues are usually negatively charged. So please pay attention and buy the correct ones. They also on the chart, they have also can be bought, but they have different other function and they're using other type of experiments. Now then it comes to multi-well chambers. We have two different, also here we have to pay attention to what we are ordering, and it depends what you want to do. There are this type of, one type of the multi-well chamber with a plastic slide at the bottom, which is also quite thick. This cannot be used, for example, for live imaging. What you do, you perform all your staining and everything in the wells, after which you remove the chamber, then you add mounting medium and a cover slip. And this is how you can go and image. And this is this type of multi-well chambers are suitable for long-term storage too. The other type of multi-well chamber has a glass or thin optical plastic at the bottom, and you should pay attention. That is number 1.5, but I think that most of them are number 1.5 or number 1.5H. The chamber cannot be removed, but you can start imaging immediately after the immunostaining, and they are very well suited for live imaging. But if you want to anyway store your sample for a longer time, you can remove the culture medium or the PBS if that was used to image your sample, and you can add some mounting medium and you can store them too, and then you seal around so it doesn't dry out. Then we continue with the multi-well plates. We have to think a little bit then we are ordering our multi-well plates. And what is important to pay attention to when it comes to multi-well plates is the skirt at the bottom of the plate. And the skirt is this plastic edge that leaves the bottom of the plate so that the glass doesn't touch really the table, then you put it on the table. But in microscopy, what we want is to work with multi-well plates with low skirt. And the reason for that is that if you have a multi-well plate with high skirt, if you try to image the outer wells, the objective might bump into the stage insert, which is not good for objective, and you will lose the focus. So please, then you see that the multi-well plate that you order has a low skirt, less than 1 millimeter. Before ordering a whole bunch of multi-well plates, ask for a free sample before ordering. So you can check some parameters because there are extra requirements when it comes to multi-well plates. As I said, they should have low skirt. They should have, the bottom should have a glass bottom number or 1.5 or optical plastic. They do not leak, and this we mean that they don't leak between the wells. It happened before that people were imaging and then they say, oh, what, I didn't put any DAPI in this well, but it was very well. We could see the staining, and that meant that the DAPI leaked from one well into the other. They do not kill the cells, and unfortunately, this is not a joke. And the reason for it is that sometimes then they might change the glue, and then they are gluing this bottom glass or plastic to the bottom of the to the well. Might be the quality or this glue might be toxic for the cells. That they are flat throughout the whole surface, and that is in particularly important when you are if you are using plastic bottom plates, because they are not super flat. That they are not the autofluorescence. This is also for optic plastic bottom carriers. So please, before ordering, ask for a sample. They're always very willing to send a sample. So you can try to go through these requirements before you order a whole bunch, because they are quite expensive. Now, another very important requirement, and something that quite a lot they don't think about, is that some of the plastic, is the optic plastic bottom, either multi-well plates or chambers might be sensitive to the immersion oil that you use, then you use an oil objective. And here you can see an image of a plastic bottom multi-well plate that was used with an oil objective. And then you can see the oil damage, reacted with the plastic, and it's not good for the object and definitely not good for your imaging. So what you should do, or the facility should basically ask, or you should ask actually, then you buy the plate, if they are compatible with the oil that you are using then, with your objective. And for the courtesy of EBD, actually, I wrote them and I asked them, do you check if your plastic is compatible with this and this objective? So I was giving the Nikon objective, and they very nicely gave me a list. And they said, yes, we checked and it works very well. So be careful, pay attention, and ask so you know exactly what you are dealing with. The sample carriers. Again, we are going to cover slips and slides. They are not clean and definitely not sterile. And here I give you an example. I totally open a new batch of cover slips, and I randomly took up 2 cover slips. And you can very nice see the impurities on the surface of the cover glass. And we don't want to image that. We don't need that. We have enough to image with our sample. So always clean your cover slip before using them. My recommendation is to use an acid-base cleaning, and for that we have made a video. We can find it on our webpage, but also on YouTube. If you are close, you have a facility near you, you can ask them if they allow you to use their plasma cleaner. It goes very fast. You can also put a bunch of cover slip, and they will clean the samples marvelously. For cover slip that you use to grow cells on, you need to sterilize them, but you do that after you clean them, either with ethanol, or I always prefer to autoclave them. So a summary, before choosing your sample carrier, which means that before even starting your experiment, it's a part of the design. You are at that stage. Think, what is your scientific question first? How I want to analyze? What do I need for resolution? And so on and so forth. Think it over, because that will help you to decide on the later stage. If our recommendation for cover slip, the bottom of the multi-well plates, dishes, and chamber is to use to be number 1.5, which means 170 micrometer thick plus minus 10. If possible, glass. If not, use the optical plastic, also number 1.5. Always check if the sample carrier you've chosen can be used with the available microscope. Check the quality of the sample carrier, because they are quite expensive, so it's better to do this before you spend a lot of money. And you should especially look that the multi-well plate is flat. Make some samples so you can check if they are leaking. And very important, that the skirt at the bottom of the multi-well plate is low and is low enough to work with your microscope. If you use thick bottom dishes or cell culture multi-well plates, if possible, use long working distance objective or extra long working distance objective. If you do not have this type of objective available on your microscope, usually the 10x works well. Sometimes the 20x, but it depends very much on the working distance of the objective, but nevertheless, it's not the ideal objective to use, or actually it's not the ideal way to image cells in this very thick plastic multi-well plates. So that was the first part of my talk, talking about the sample carriers. Let's stretch a little bit, take a little break. 1-2 minutes, so maybe go take a quick coffee and come back.
Gabi, can you just say clearly which time they should come back?
What time is it?
Look at your computer. It's 10 past.
Okay, in 5 minutes.
Okay, 15 past. We come back. Thank you.
Okay, so let's continue. We will continue with the next topic. Where should I place my sample? So we already heard before, the sample should be directly on the cover slip, and the cover slip should be number 1.5, after which we will add our multi-medium, our slide, our supportive sample carrier, and we go to the microscope and we place our sample on the stage, and we want to image, and this will how the setup will look like. So both the excitation light will reach directly the sample, and the emission coming from the sample will pass the objective and reach your detector without any of the multi-medium interfering. So we don't have any light scattering. Also means that more light will reach the samples, so our samples will be brighter, it will be easy to focus, our images will be sharper, and we will get a lot of our data will be reproducible. The question is, do we always manage to have this sample setup, this perfect setup for imaging? So let's go to the cells, usually monolayer, thin samples, monolayer, or adherent cells. We grow them directly on the multi-well plate, if that's what we use, or dishes, or the multi-well chambers, or the cover slip. So whatever we do, the cells are directly on the glass. If we have cover slip, we grow them, and after we do all the immunostaining with them, needed, we take the slide, we add a little multi-medium and flip them over, and they're also ready for microscopy. So in all these cases, we have the best imaging setup for our sample. But what about cells on suspension? And quite a few we also want to image them. We cannot just image them, growing them in the multi-well plate without an extra step that we need, because they're moving all the time. So quite often, what we do, we are using a cytospin. And here I just depict one type of cytospin. When what we do, we have a slide holder here, and then we have a tiny little holder where we will add our cells, and we take the slide and we put the slide here in the slide holder. We add the cells and we give it a good spin. And what will happen is that the cells will end up on the slide. After that, we do all our immunostaining steps, and we add a drop of multi-medium, and we add our cover slip on top, of course, so we can image. So if we do that, this is how our sample will look like if we look at the side and we are at the microscope. Now, a few things change, because the sample is not anymore on the cover slip, it's actually on the slide. So what will happen? The light that will reach the sample will have to pass through a thick layer, or the layer of the multimedia that you have. So that will reduce light scattering. Less light will reach the sample, so the sample will be dimmer, as we already we will see. It will be not sometimes it will be harder to focus, so less sharp images, and we will also have some irreproducibility in our data, because depending on how much multi-medium you add one time or the other, well, that will influence both the focus, the light, and that will also read that the data might not be so reproducible. So what can you do? So what can we do if we have these cells in suspension? Can we change this? Can we in some way make things so that our cells end up on the cover slip? Yes, we can. So we still use our cytospin, but what we do, that you take your slide, and on top of the slide, you put your cover slip, and then you fix it there with a tape. And this setup you put here on the slide holder. So now, when you are spinning, the cells will end up on the cover slip and not on the slide. After that, you will do your immunostaining. When you are done with the immunostaining, you remove the tape, you gently take off the cover slip, You add, again, a drop of multimedia on the slide where the cover slip was. You take your cover slip and you flip it over. So now, basically, you have your cells on the cover slip, and again, you ended up by having the perfect sample for microscopy. Just a little tip. But about tissue sections. Well, what we always do, or most of us do, or they have to do, we don't have a choice. We take our slide and we are very careful to use the correct type of slide, which are charged, so our tissue will stick to it very well. We go and we place our tissue section on the slide. We perform our immunostaining, and we go, we take our bottle with multimedia and we squeeze out a drop, and we cover with the cover slip. So again, this is how our setup will look like. So again, our tissue is on the slide, so it's far away from the cover slip. We will have light scattering and dimmer samples, less sharp images, and reproducibility of the data. And this irreproducibility comes mainly for this amount of, also among other things, amount of mounting medium that you add. One time you will squeeze out a little bit, the next time a little bit more. It's very hard to do it right like that. But can we minimize this light scattering? What can we do? First, you add, as I said, as little or you heard before, you should add as little mounting medium as possible. Because the goal is here to add just enough mounting medium to completely cover the sample and fill the space under the cover slip. What we don't want is, what we want is to create a thin, even layer without the cover slip floating all around, or then we add, we can see that it comes out, seeps out from under the cover slip. We don't want that. So what we have to do is to measure and find the minimum amount of mounting medium that covers our sample. As I said, every time we squeeze, we will squeeze different amount. I know because I tried it. It's almost impossible to squeeze always the same amount, especially if you have these very large areas and cover slip you want to use. So the distance will all the time be vary from one sample to the other. So what you do, if you measure, you will always, you might not be, the cover slip might not be very close, or the sample not very close to the cover slip, but you will do your best to minimize that this distance. Another tip I have is to use smaller cover slip. Don't try to squeeze so many tissues on one slide. You know, it's okay to only have one or two. You might have, okay, 2, 3 extra slides to work with, but it's much easier to measure the amount and to find the minimum amount if you are using smaller cover slips. So just an idea and a tip. Here, to prove you how much the amount of the mounting medium plays a role in your imaging, what I did, I created this kind of sample. So I had cells on the cover slip. I grew them on a round, nice round cover slip. I stained them with phalloidine fits by the book, exactly as according to the protocol. And then I was done. I took 2 large cover slip, also number 1.5. And on one of them, I added 5 microliter mounting medium and the other 20 microliter. And then I flipped over the cells on these slides. So this is how my setup looks like for the first sample, and this is how it looks like for the second sample. So idea was that to image them from both sides. It was not easy, but it happened. So I took first the first sample, and again what I did, I make the same sampler, I put it on the stage. Now I'm imaging through when the sample is closed, it's on the cover slip. I always did my best to find the best focus. I used the same imaging settings for imaging, and I have the same display to show you the images, and I use the 20x AR objective. So first I took the image with the sample on the cover slip, then I flipped over and I tried to find the same cells, and I imaged through the 5 microliter mounting medium. Although we already said, and Sylvie mentioned it several times, don't believe your eyes. It's the data, the measurements that matter. If you just take a first peek at the images, you would say, yeah, well, they look quite the same. You know, I cannot really see a difference in the brightness. But again, we don't always believe our eyes. So what I did, again, I tried to find a dot, something that was common for all of for both of them. I find it there, and I look at the intensity, how it looks like, and the measured intensity. And you can see that I always have, it seems that it's exactly the same. It's very little different. So they have the same brightness, the two images. And if I zoom in and I would try to find some details on these cells like Philopodia, I can see them quite well in both samples. When the sample is on the cover slip or when I image through the multimedia. But the amount of media was so little that I really created a very, very thin layer. Now I took the other sample and I have 20 microliter multimedium. And again, I tried to do my best to focus, same imaging settings, same display, still using the 20x AR objective. And the first image goes, and I image with the sample on the cover slip. And then I flip over and I took another image. And now I'm imaging through the 20 microliter multimedium. And just looking at the image, and I said, I try to do the best with the focus, and I need to change the imaging settings. This is how my image look like. And it's quite obvious that the brightness is actually changed. And on top of that, and I try to zoom in and try to find some very fine structures. It was not a problem to find it when I was imaging. The sample was directly on the cover slip, and my objective was coming close to the sample. In comparison, when I was imaging through the 20 microliter mounting medium, the detail were lost and the image is less bright. And this was the AR objective. So let's see what happens if we are imaging using an oil objective, and I do the 60x oil objective. Again, first time I would image, I took the sample with the very little mounting medium. I tried to do the best focus, same imaging settings, same display, and this is how the image looked like. Already here, I can see that I was not able to focus. So this was the first image, of course, I took, and then I flipped over, and I tried to take another image of the same cells, and whatever I did, I was not always, it was very hard to find exactly the same focus already here. But the brightness of the image, it's the same. Now, what happens, now I take the other sample, the one that I had a lot of mounting medium, 20 micrometer, and I do the same. Take the first image, and the sample is on the cover slip. Looks very nice, you can see very well the details, and then I flipped it over, and I image through now a lot of multimedia. And this is how the image looks like. Much less bright, so, and I have a very hard time to focus and get a very sharp image. So the high NA objective are they're very sensitive to the if they are very sensitive for when imaging, and you have a lot of multi-medium between the cover slip and the sample. So add as little mounting medium as possible. Now, we can also place another option. Now we realize that, yeah, if you have your sample on the slide, the best you can do to minimize all the problems that comes with the sample being far away from the cover slip is one way is to minimize, to add as little mounting medium as possible. Another way to solve this problem of light scattering and so on is to place the tissue section on the cover slip instead of the slide. Well, to place your tissue sample on the cover slip instead of the slide follows the same procedure as when you are doing it, you are placing it on the slide. And we have a very nice video on our homepage how this is done. We actually film a person working at the facility who is doing cutting and sectioning, and he did it for us, so you can have a look how it's done. A tip for all of you, if you want to try to do that, is since the cover slip we usually use the big rectangular cover slip, and they're very thin, so it's not very convenient to hold them, and you will always be afraid, I will break them. What you can do is to put them on the slide and fasten them there with the tape. And now you are holding the slide, but then you will cut or give to the technician to do it. Anyway, the tissue will end up on the cover slip. And as I said, it's not hard to place the tissue section on the cover slip, but to keep it on the cover slip is not always easy, because if not coated, this cover slip have no charge. And there are certain tissue types like bone cartilage or fatty tissue that are notoriously known to detach easily, sometimes even for the charge slides. On top of that, if you need antigen retrieval, then it might happen that the sample, you will lose your sample. But what can you do something to improve the attachment of your tissue to the cover slip? Well, what we can do is to do something about on the cover slip slide. So usually what we recommend is to perform a harsh acid or base cleaning. And I also, what you do actually, you are ruffling the surface of the cover slip. And then will enhance the attachment of your tissue on the cover slip. Another option is to coat the surface with adhesive, adhesive that will create a positive charge. And I suggest to do that after you clean your cover slip with acid, or you perform an acid or base cleaning. And there are many different type of adhesive you can use that will have a positive charge and will enhance the binding of your tissue, which is usually negative, and it will keep it on the cover slip. If you have tissue sections that are thicker than 40 micrometer, I suggest that you perform your immunostaining or all the steps that you need in a free flow, so-called free floating, in a tube or in a little well, or whatever you prefer. And so you do everything there, and then you are done. Then you very carefully fish your sample and you put it on the cover slip. Ideally, you can add another cover slip on top, like a sandwich, and then you can even image it from both sides. What about organoids, islets, and even thicker tissue sections? This will, since they are so big, they will fall off the slide or the cover slip. But there, of course, we want to image them too. So what can you do? Well, the one way is to use spacers that are, they are sticky on both sides, and the spacer are not only they are sticky on the both sides, but they are also have different thicknesses. So you actually make a little chamber. We will create a little chamber where you can put your sample, and you can find in different shapes and different sizes. So my suggestion is always try to find something which is closest to the size of your sample. So what you do is that you take the cover slip. Now you have your spacer. You remove one sticky side and you put it on the cover slip. Now you have a tiny little chamber. This is not at the scale, but the image. And then what you do, you add a tiny little bit of, you add your sample. Then you need to immobilize your sample with something, usually, so it doesn't move. So we suggest low melting point agarose or acrylamide. And then you will add your clearing medium. We are talking of big samples that we need to clear so we can image through them. And the idea is that you will clear the sample and your whatever you use to immobilize your sample at the same time. So you add your clearing medium. Now you remove the second sticky part of your spacer. And what I suggest always to put another cover slip number 1.5, so the or number 1.5, or if you don't want that or you find this too troublesome, you can add the slide on top of it. So this is a very elegant solution to image organoids, either big samples. I can recommend to look to this video. It's on YouTube where they actually show a little bit one type of this spacer, how to work it and how to handle the spacer. Of course, you can also use a multi-well place or chambers to do the same thing. You will need much more clearing material. Nevertheless, you always think about that you have to immobilize your sample in some way because we don't want the sample to move every time we move the stage. So now, if you look to this sample, can anyone tell me what's wrong with this slide? Can anyone tell me what's wrong with this slide?
The cover slip is very close to the edge.
Yes, exactly. The cover slip is very, very close to the edge. Very good. Did you ever, before the course, actually think about it? That it might be a problem?
Yeah.
Good. Well, now I will, for those who might haven't think about it, explain why this is a problem. Because indeed, what happens that if you prepare a sample like this is that the cover slip is very close to one edge. So then you put your sample on the stage. It will rest on one side. It will rest on the cover slip plus slide. And on the other side, like here on the left side, here only on the slide. And the sample will not be flat. And if you will try to focus, you will always have the impression, what's happening? I really focused, but if I'm always in one side out of focus, that should give you a very good clue that the sample is not flat. So what you can do is, of course, to move it. And it's totally okay to end up on the white part of the slide. That's just there for you to take some notes, to write some things there so you know what the sample it is. And you will be able to read it anyway. So if you move a little bit, now then you are placing your sample on the stage, it will rest on the slide on both sides and your sample will be flat. A little guide for you is that you buy your slides and you have the little plus, which actually denotes that it's a positively charged slide. Always have that as a guideline and always move it left to that from the plus, and that will be good enough. And it will rest flat on the stage insert. What else I observe sometimes when our user come to us to image when I do the training is that quite a few of us want to do everything by the book. So you also use these sticky labels so where you know what the sample is, or especially if you got the sample from another facility who cuts your sample, they are very good on putting a label. which is okay. The problem is that they put the label quite often on the same side where your sample is. So that means that again, then you are putting your sample on the stage, it will you will have the same problem. In one side, it will rest on the label plus the label plus the slide, and the other only on the slide. So please, if you do that, always put the sticky label only on the other side from where the sample is, and then everything will be okay. Okay. So in summary, if possible, always put your sample on the cover slip. Oh, sorry. Add enough mounting medium to completely cover the sample and fill the space under the cover slip. Do not cover the ends of the slides so that you can keep the sample flat on the microscope stage. And let's take a 10 minutes break. So we will be back at 10:50. Okay?
Great. Thank you, Gabi. And I guess if there are questions, people can ask also.
Yeah, that's the idea that we have. If I finish that we have 30 minutes of question now, they can.
Yeah. Okay.
Yes. But luckily they heard already a little bit.
Yeah.
But it's
I think it's good. There's lots of small details, so it's good to hear them several times.
Anything that I left out, but I think that most of them are covered.
No, I really like the images that you took now to demonstrate the stuff. This is so powerful, I think. Very good. Okay, I go for a break.
Okay, so let's continue. And now I will move to the next topic. What should I use to fix my sample? So the fixatives are super, super important. And most importantly, we have to take care of them and handle them properly. The main goal of fixation is to preserve the cellular morphology, integrity, and the structure of your sample. The fixatives can be grouped in either as cross-linking agents or organic solvents. Among the cross-linking agents, the most commonly used are paraformaldehyde, formalin, or glutaraldehyde solution. As organic solvents, the most commonly used are acetone, methanol, and ethanol. But what is the difference between them? The difference between them is the way they interact with the sample. The cross-linking agents form cross-linking bridges between the proteins. They are then used, they preserve very well the cell and tissue structure. The fixation is usually performed at room temperature, but it's a very slow process. It can take from minutes to hours, depending on the size of your sample. And especially if you want to look at your target is within the cell, it requires permeabilization. You need to use a permeabilizer after the fixation. The organic solvents, they act by dissolving lipids, they dehydrate the sample, and they precipitate the proteins. They are much harsher on the cells and tissue structure. Usually the fixation is performed on ice with ice-cold solutions. It's very fast, usually between 5 to 6, a few minutes, 6, 7 minutes, not more than that. And you do not need to permeabilize your sample after using these fixatives. As I said, the most commonly used the cross-linking agents are paraformaldehyde solution and formalin solution, and I will spend a little time to talk about them because they are there are some things that you always have to think about when using them, and also a few words about methanol. Again, paraformaldehyde and formalin. There is a difference between them, but the solutions, I would say. So the paraformaldehyde it's a white powder that in order to use paraformaldehyde as a fixative, first you have to make it, to heat it up in PBS, quite high temperature, 60 to 70 degrees to make the solution itself, and that can be used as a fixative. And the most common concentration used of paraformaldehyde that is used as a fixative is 4%. But formalin, on the other hand, is something that you already buy. It's ready to use. It's an aqueous solution, depending on the manufacturer or vendor, 37 to 40% formaldehyde. So it's basically the gas. Formaldehyde is a gas that is dissolved in water, and you make the formalin. And most important to keep in mind that it contains methanol as a stabilizer, 10 to 12 or 15%. What you use as a fixative is a 10% formalin solution. That corresponds more or less to 4% PFA. So it's important to make this distinction between the two solutions. Sometimes you can also use sealed ampoules, which you can buy of various concentration, 8 to 16 or 20%, and usually they are methanol free, they are hermetically sealed, vacuum sealed, so they are usually methanol free, but you have to check. So what should we think about when we use, or how should we handle these two solutions? So if you use a paraformaldehyde solution as a fixative, you will prepare it yourself from powder. Always make a stock solution. We recommend an 8% stock solution with the correct adjusted pH. You really need to adjust the pH so it's between 6.9, 7.12, so it should be neutral. Anyway, the solutions always have a tendency to go back to be more acidic due to oxidation. Then you make this solution, let it cool, aliquot it in 50 milliliter vacuum tube and freeze down. When you defrost the solution, and you use it, the defrosted solution can be used I would say max to 1 month if kept in the fridge. Always write the date on the tube that you defrost it, so you know the validity, and you say, yeah, well, I can still use it, or no, I have to take out another layer coat. If you use the formalin solution as a fixative, always keep the concentrated Stockholm solution, that is the 37–40% at room temperature, well sealed and out of sunlight. Once you open this stock formalin solution, you should not use it after more than 6 months, because in time it will oxidize and it will affect also the pH. And the same with the 10% formalin solution, which are already the diluted one, have even a greater chance for oxidation and pH drop than the stock solution. So them you should not use longer than 3 months after the initial mix. Again, as I mentioned, the most commercial formalin solution might contain methanol to stabilize the solution, and that you have to check if your antibody is actually compatible with methanol, meaning with the way the methanol will affect also your fixation. Okay, so these are some important features to keep in mind how to deal with your fixative. The fixation time, it's very critical. You don't want to overfix because it will introduce artifacts, or and it might mess the epitope, which will require then a harsher and longer antigen retrieval step. And we might want to avoid that as much as possible, if possible. But you don't want to underfix either, because it will lead to the degradation of the target. So you have to adapt the time, temperature, and concentration to the thickness of the sample, especially when it comes to tissues. If you fix the fixation of cytoskeletal structure, can also benefit if you warm the fixative, 37 degrees. So you prevent a cold shock to the cells and thus alter or disassemble the cytoskeletal structures. So a tip, we are doing it. You warm up only as much as amount of fixative as you need, and then you take out your sample, carry your cells, and you add your fixative directly to the culture medium, volume to volume, to leave it 8 to 15 minutes, especially if the cells are very thin, so they don't need a tremendous amount of time for fixation. But of course, there are exceptions depending on the cell type. Either you give this at room temperature, you can put it back in the incubator, and after that, you rinse gently with the PBS. Just a tip. As I said, a few words about methanol, acetone, and ethanol. Sometimes methanol can also be used by itself as a fixative, but it's quite often you will make a solution, for example, acetone, ethanol, or different concentration, and between them, they are always used as pre-chilled or ice-cold solution. They have a very short, usually a short fixation time, and you should not use this fixative if you want to look at the membrane-bound epitopes. So much about the fixation. Let's move further to autofluorescence. And you already heard a little bit about autofluorescence. And autofluorescence is the major source of fluorescent background in biological samples. And we have several sources for autofluorescence. The most common source comes from your sample itself, or basically the sample containing common endogenous fluorophores like NADPH, flavin, lipofuscin, porphyrins, and you name it, collagen, elastin. Another source is the mounting medium. And before I move on, here you can see an example of the spectrum for all these endogenous fluorophores, the emission spectrum after they were excited with a 366 nanometer wavelength excitation light, and you can see that the emission spectrum for these fluorophores, they are covering all the visible spectrum. It's actually all the spectrum that we want to image within. So that if we have autofluorescence, we will see it in our when we are imaging. And as I said, another source for autofluorescence is the mounting medium. The cell culture medium can be autofluorescence, the bottom of your carrier, if it's not glass, and the fixative can also give rise to autofluorescence. Here it's an example from a brain tissue that was imaged, no staining, no anything, just the image in the green channel. And you can see the signals that these come from lipofuscin, which is the most common molecule to give autofluorescence. So, and these signals look very, very specific. Sometimes they only show up in one cell type, but they can also show up as a general background signal. So then working with a new tissue, always start with imaging unstained tissues to understand in which channel, if you want to understand if you have autofluorescence, and in which channel the autofluorescence is the problem. Sometimes if you find in which channel is a problem, you might want to move your fluorophore on a totally different channel. And when you are you work with your sample, the so-called no primary, no antibody, we'll talk a little bit later about it. Your sample should be treated exactly, then you prepare this sample exactly as your main sample. But how to reduce the unwanted fluorescence? There are chemical treatments, and here I just list a few of them. The most commonly used, Sudan black, black B, TrueView, copper sulfate, or natrium borohydride. These are the most commonly used. But you have to be aware that then you will use them. They might affect the immunofluorescence labeling. So sometimes you will have to make a compromise between autofluorescence reduction and antigen visualization. Some treatment will work better on one type of tissue, but less on the other. So some treatment need to be applied before. Some treatments need to be applied before you start the immunostaining, and some of them after the immunostaining. There are also so-called non-chemical treatments like UV radiation. They usually take a longer time, but the time is depending on the power of your UV light source, and they are quite often used in combination with other treatments. Another way to get rid of the autofluorescence is to use a spectral detector to separate the autofluorescence spectrum from the spectrum of the fluorophore used to stain the tissue. And of course, knowing in which channel your autofluorescence it's a problem, you might want to use to move to another channel. So you can switch from green, for example, to red or far red or near infrared fluorophores. Always check the literature. You don't have to reinvent the wheel. For most common tissue types, there is already something there in the literature that can get you started, so you have a starting point. And here is an example how Sudan black applied to a spinal cord tissue reduced the autofluorescence. But as you can see, you have to make a dilution and titration to find out which one actually works for the best concentration, works best for your tissue. As I said, autofluorescence can come from the mounting medium, and because mounting media degrades in time, so you should always store it according to manufacturing instruction, but it will degrade, especially if a lot of people are using the same mounting medium. So you should always check regularly if your mounting medium is autofluorescence, and how to do it is very, very simple. You take a drop of mounting medium, you put it on the slide, and you cover it with the cover slip. You make some bubbles, and what you do, you go and focus in the bright field. You find your bubble, and then you go over to fluorescence, and now you take images in all your favorite channels, the one that you actually use, then you are imaging your real sample. And if you see a dark, your bubble dark with a dark inside, and the outside you see some fluorescence, that means that your mounting medium is autofluorescence. So what you should do is basically buy a new one. As I said, the multi-medium can come also for the culture media, especially for those who are imaging live cells. They have to pay attention. So you should always use an imaging medium with no or very low flavins or vitamin B12 or phenol red. They give autofluorescence. And here is an example. Then we measure the background of our sample with cells which were in the MEM with phenol red and the other only with PBS. And you can see that the background doubled up. And especially if you want your sample is very dim, your signal is very dim, you don't want it to drown in the background coming from the autofluorescence. Always keep your imaging medium in the dark in the fridge, because a lot of components are very sensitive to light. They will degrade, and in turn, they will give rise to autofluorescence. At the bottom of your sample carrier, if it's not glass, might also give rise to autofluorescence. The fixative, aldehyde or glutaraldehyde. So avoid that. This is because of the reaction they have in the sample with your different components. So if possible, avoid using glutaraldehyde unless there is a very, very specific reason in your protocol to use it. Fix for the minimum amount of time required for the size and type of your sample, and do not use solutions that are old or stored in warm condition or have been in contact with air for a very long time, especially, for example, bottles which are not closed properly, and always wash thoroughly after fixation. We take a 5 minutes break and we come back. What is it? 11, I would say 15. Okay.
I have a question. Should I make it now or at the end of the lecture?
Well, we can take it now if we have this little break.
Okay, so you mentioned that we should buy the powder of PFA and dilute it and then make aliquots, but what if we buy already the diluted PFA? So in the lab we use a 4% already diluted PFA.
Well, it depends how you bought it as a 4%. You bought it as an ampule or
4% we always use.
But in ampules, which are sealed, or
no, it comes like a big bottle already.
Well, you know, that's the problem. You bought a big bottle that everybody opens, close, open, close, and it's already diluted. So that what you can do at least is to measure the pH. You know, because the diluted solution have much higher tendency to oxidize and drop the pH. And then it's not something that you want, because all these properties of the PFA, which is a long chain of, as that, then we make a solution, we cut it off, so it actually can act as a fixative. And then we alter that. So buying this already diluted solution is not the best. Or then you have to take care, if you buy next time, aliquot it yourself, you know, take and freeze it down, and then, you know, always take up exactly as much as you want. And, you know, to measure the pH, you take this paper, pH paper. Oh, it's okay, it's still neutral. You don't want to, so it drops to more acidic. That's not good.
Okay. Thank you.
You're welcome.
I also had a quick question. Is there any visual cues that you can recognize that for overfixing?
For overfixing, well, usually the cues are that, especially if you have a cell monolayer, that where in general you don't have to perform antigen retrieval, that your antibody is not working as it should. You know, maybe it will work before. I say, oh, what happened? I used it before, it doesn't work now. And you check everything. Yeah, I follow the protocol exactly, but maybe you overfixed your sample, and then can be a way. Or if you get exactly what I said, too much autofluorescence, that is also a cue that something it's wrong.
Okay, thanks.
Might be 3 minutes late.
Sorry?
I might be 3 minutes late.
Yeah, but it's fine. We have plenty of time, so it's quite flexible.
Blah, 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 as fast as I could.
No. Please don't blah blah as fast as you could. We have plenty of time. Now we have more time, right? So compared to before, so it's good.
Yeah, that's very, very grateful.
Yeah.
But yeah.
Did you say 15 past? People come back.
Yeah.
Yeah? Okay.
I thought it was a very heavy
Yeah, no,
part.
yeah, I think it's good to take
Right.
breaks. Yeah. Good. Thank you.
Hi, Gabby. Can I ask a quick question about the mounting medium?
Absolutely.
Yes. When you add the mounting medium to your sample, do you usually do you need to press out uh, do you need to press out the cover slip so that the mounting medium covers the sample, or?
I mean, if you work with cover slip, then you usually have them in a, let's say, 24 well plate, isn't it? And you dump there this nice round cover slip. Or
Uh,
that's what we are doing. Or how?
yeah, on the slide.
On the slide. Well, on the slide. Well,
Cover slip and slide, sorry.
so you have a slide, you have your sample, and you have usually your PET pen around that. Okay, so you add your the fixative and you leave it there for a certain amount of time, depending. It depends how you prepare the sample, and we will talk about little later, because this fixation, maybe you fix it before, maybe you fix it after, but usually you just leave it there. And if it's a very thin surface, it's a thin tissue or a thin sample, it's totally okay to leave it like this for a few minutes. So it's totally okay, and then you wash it up and you continue with your immunostaining. This was your question?
No, sorry, it's about mounting the
That we will
mounting.
come right now after this.
Okay, okay, thank you.
I will talk about multi-medium exactly after now.
Okay, yeah.
Okay, so let's get started. I'm a little bit late. And okay. So a question to all of you. How many of you checked before the course what is the refractive index of the multi-medium you use? How many of you actually effectively checked and looked up? Anyone? Ooh. So before the course. Super. That's good that all of you checked and knew exactly what is the refractive index of your multi-medium before the course. But anyway, let's wrap it that up. What is all this multi-medium about? So the role of the multi-medium is to protect the sample from drying out. It will prevent the sample from moving, and it will increase your fluorophore stability because the solution contains anti-fading agents. And most importantly, also, it will homogenize the refractive index for microscopy, thus it will reduce light scattering and spherical aberration. The question is, are all the multi-medium the same? No, we have two categories. We have water-based and organic solvent-based. Within the water-based category, we have non-hardening, which sometimes you will also find the word non-curing or soft setting, multi-medium, or hardening / curing or hard setting. Depends on the manufacturer. What is the difference between them? The non-hardening will always remain liquid, and it has a refractive index between 1.4 to 1.49. You will always require sealing, so you have to seal after your cover slip, around your cover slip. You can use nail polish or wax. But the good thing is that the cover slip can be removed. And the backside is that you can your samples will be preserved for a shorter time. So again, you put your sample, you put the multimedia, and then you have to seal it all around. Then you have, but you can start imaging as soon as the nail polish is dry. If you have a hardening multimedium, then it will solidify. The refractive index is between 1.47 to 1.52. No sealing is needed, so you should not seal your sample, because otherwise it will never solidify. But you need to wait quite a long time till this multimedia will solidify and reach the final refractive index that it's written in the data sheet. But you can preserve for samples for a long time. So please do not seal your sample. You can start imaging when the multimedium harden and reach the refractive index that was specified. Then we have the organic solvents. They are always hardening marking medium, but you need an extra step of the hydration. Then you use this multi-medium, and they also always have a hard refractive index over 1.5. Again, no sealing is needed, but it takes a long time for this multi-medium to solidify, but you can preserve your samples for a very long time. So the choice of your multi-medium is largely dependent on the sample time, how you will analyze your sample to answer your scientific question, and the type of application. If you are doing immunofluorescence or immunohistochemistry, there are special multimedia that work best for histocytochemistry that you should work with, because they will preserve the color much better. So if you should use a non-hardening multimedia if you are imaging live samples, and by this I mean because the culture medium is also multi-medium. and you should use correspondingly the one that suits you, your cells. If you want to be able to remove the cover slip, for example, to run another round of staining, this is when you choose a non-hardening mounting medium. If you need to measure distances or volumes in 3D, if you have a fixed sample, there are special non-hardening mounting media with optical clearing properties. As example here, slow fade glass. When you clear big samples, because the clearing medium is also a mounting medium. And keep in mind, if you do not need to preserve your sample for a very long time, you should use a hardening mounting medium if drying or shrinking of the sample are not an issue, because of course the hardening medium might a little bit shrink your sample. If the refractive index of the sample and the objective immersion medium is around 1.5, if you need to preserve your samples for a long time. But please do not use any mounting media with DAPI on it, because that will increase your background fluorescence. It's for sure the DAPI binds very well to the DNA, but also remember that this emission spectra is very wide, so you will see it you might see it in all the channel. Nevertheless, your background fluorescence will increase. So please, no DAPI in your mounting medium. DAPI should be used at the last step of your immunostaining, either together with the secondary or after that for 5 minutes and you are done. Here it's an example of different type of mounting medium. And you can see the arrow, the two mounting media, which are hardening mounting media, and you can see how they shrink the sample. So please be aware, think, what do I need to analyze? How do I want to analyze my images? And then think in advance which mounting media you should add. Well, what should you pay attention then you mount your sample with your mount your sample before you add the mounting media? Be sure that you remove all the PBS to avoid diluting your mounting medium. But of course, think about that you don't want to leave your sample to dry. Drying a sample is not a good idea. If you use a hardening, always allow it a hardening mounting medium, allow it to fully harden on a flat surface before imaging. So really it totally hardened, not after 3-4 hours. Oh, I take it and I put it in the rack, which are when it will be set for horizontal, it will be now vertically, because if it's not fully hardened, you will get a tilt in your sample. Again, if you use a non-hardening mounting, you have to completely seal the cover slip. That first of all, to avoid that it will dry out, or it will the multimedia will come in contact with the objective, and you can start imaging only when the nail polish is completely dry. Just one tip for you: then you add the nail polish. Please do not add as much that you are minimizing your area what you want to actually image. Just add as little as possible, so you still preserve a big area for imaging. Can't you remove the mounted cover slip? Yes, you can. If you use a non-hardening mounting medium, what you do is that you you take a sharp blade and you cut here around where you know that the cover slip ends. You cut around gently, and then you take the slice and the cover slip and you place it in PBS, room temperature, and very gently, under gentle agitation, some 30 minutes. After the 30 minutes, you will see that the cover slip is floating. And then, of course, depending where your sample was or not, then you can rinse with extra PBS and start the next round of staining. Also, before you go to imaging, quite in a few samples, you can see, especially if you did all your staining on the cover slip, you have your sample on the cover slip, you could see a very white haze on the top of your cover slip. and that are PBS crystals, and you should remove them before you go and you image, because it's no idea to image through a thin layer of crystal. And on top of that, you don't want the crystal to interact with the oil if you will use an oil objective. And to remove it, it's very easy. We just add a drop of water and then you gently clean it up. You swap with the microscope, the paper, and you add another drop, and you do it again. Basically, you have to do the same to clean your sample. If you are using oil or silicone or glycerol or water, and the way to clean your sample, it's the same way as you would clean an objective. We have this, we recommend this nice video, and you of how what they show how to use to clean the objective, and you should use the same technique there to clean your sample. The most important points then: preparing your sample for microscopy. Choose a sample carrier adapted to your experiment. Always use cover slip of thickness specified at number 1.5. Whenever possible, place the sample on the cover slip, not on the slide. Use fresh fixative solution when fixing your sample. Do not cover the edge of the slide with the cover slip. Check regularly the autofluorescence in your mounting medium. Always allow the mounting medium to completely harden. If you use a non-hardening mounting medium, use nail polish to completely seal the cover slip. Do not use mounting medium with DAPI. And I think that's it for me.
Good. Very good. So I think it's quite a lot of things. I hope that you guys have you got some tips and wrote them down, and so that you can so that it's possible for you to write your assignments and summarize later. So Gabi, thank you very much for this. There is another lecture in the afternoon about immunostainings, more specifically. And now we have a group discussion, how to improve your sample preparation. But before we do that, I'd like to know if there are some questions also for Gabi.
Yes, I have a question about the mounting medium.
Mhm.
So I'm doing live imaging and using a homemade imaging buffer. It contains glucose, 10 millimole, some HAPs for buffering, and some cation ions, and some salts.
Yeah, I saw that in your assignment, yes.
And I would like to know if this can be autofluorescent.
I doubt it, that it is. I don't think so, because I cannot see any particular components in it at all. So I wouldn't say this is, as you said, a homemade, so it does not have any of the more classical chemicals that are in the regular cell culture media that usually give rise to autofluorescence. So I don't think so. And as I also said, we can measure exactly the refractive, what is the refractive index when you come for your imaging challenge. So you have an idea, where you are. Probably is, as you said, around 1.33 or something.
Okay, thank you.
I also have a question with the mounting media. I use prolonged gold. And I mean, as you said, there are two problems because it comes with these dropper bottles. So one is that it always is a different amount. And the second is that it usually makes a lot of bubbles with this dropping bottle. Do you have any advice? Do you usually put it in an Eppendorf tube and then
Exactly.
pipette it? Or
That's the easiest thing to do. You just put it, drop, and then already knowing. So you can try. Okay, all are the glycerol base. So actually you can even read in the composition. So you can try how much you need, and then if you have it in the tube, you cut a little bit the tip of your tip, so you can always take up exactly the amount. But then you can measure. because with the squeezing out, it's a nightmare. It's never the same amount. I tried, and so one after the other, trying to be the same, but it's not. And then the distance will be different. So no, so that's the best tip I have. Put in an append or two. If you don't use it up, put a aluminum foil around and keep it exactly as you will keep your main bottle. And that's it. And then
Okay.
it's done. It's done. It's finished. You take a new one. You squeeze out a new one.
All right.
It's very easy.
Thanks.
I have a question. If I'm using PBS as imaging media and I want also to reduce the phototoxicity, and should I add something to the PBS, or because I cannot use imaging multimedia because the refraction index is different?
Well, it's their lifestyle, so it's not the phototoxicity. Where do you think it comes from? It comes from your imaging.
Yes.
So there are some chemicals you can buy, and you could mix it in your then you are imaging. So yes, there are the same components that they are added in the multi-medium that you buy. That can also be bought. I don't know now, remember from her, but I can find out which are these components that they will help to reduce the phototoxicity, and then you can make and add it to your solution to the same concentration. So it's totally possible to do
You mean
that.
in live samples or?
No, it's fixed
No,
samples.
it's fixed. Okay, right.
But I'm afraid of quenching, like if I imaging for so long.
Quenching,
Yeah.
In bleaching.
Oh yeah, bleaching. Yes.
bleaching.
Yes.
Yeah, but that's it's always a problem, the bleaching, and especially if you are using PBS. So if you absolutely need or you have to use PBS of some reason, then you can add some anti-fading
Anti-fade.
anti-fade
We will
agents.
talk about the bleaching later in the course, and we will have a little workship about that so that you realize what it is.
I mean,
Great.
then I started 20 years ago. I was actually preparing, adding some anti-fading agents to my solution. So I don't know it from top of the heart, but I can find out that it's the same that they are putting on the multi-medium.
Okay, thank you.
Okay, I think we have to move on to go to the group discussion.
But I will pop in the group, so.
Yeah, yes, absolutely. Yeah, exactly. That would be great. Okay, so everyone, we go to the Zoom. Ciao, see you there.